we're interested in protein evolution. We have been for a few years, and from different sources of material and studies and simulations, it seems that not all 20 amino acids have been here throughout cellular evolution. It seems that the most ancient proteins were composed of only their subset. We call them the early amino acids. They're generally the smaller ones, the simple ones. And these simpler amino acids were probably here since the prebiotic Earth environment. The more complex late amino acids, on the other hand, seem to require biosynthesis, so some level of metabolism already around. Okay, and I know why I have to click this. The early amino acids, interestingly, are still the more abundant ones in today's proteins. They form roughly 2/3 of the current proteome. Not sure about mycoplasma, have to check. The late amino acids at the same time are the more sort of energetically costly. So there surely is some sort of selection on keeping them less abundant. So the questions that we have been asking is, okay, do we actually need all 20? And we have so far been doing our work mostly in vitro. So looking at individual proteins out of cells, looking at the protein biochemistry when we reduce the pellets of 20 amino acids into a smaller pellet of the 10 amino acids. It's interesting that most of the amino acids that are important either for catalysis or for binding are among the late ones. So that has been the challenge that we have been facing. And I'm going to show you two examples as a proof of concept that yes, we don't need all 20. The first one is something we played around with an RNA binding protein, C-terminal domain of a ribosomal protein. I'm not going to go into detail here. I don't think you really care the methodology, but the simple message is we basically randomized the sequence of this small domain, replacing all the late amino acids, all the positively charged amino acids, which is interesting, to the early amino acid options. And we selected a variant using an RNA display, still having the RNA binding capacity and only composed of the 10 early amino acids. So not only we don't need all 20, but we can do with just half of them. But of course, this is totally in vitro, one example of a protein. In this case, you could wonder, how does this interaction work? And this way, there was just more involvement of metal ions, which quite nicely fits with the sort of origins of life scenario. We also looked at one enzyme. It was the dephosphocoenzyme A kinase. And this one, we replaced all the aromatic amino acids, which we believe were really important for the structural integrity of this protein. And true enough, the protein was kind of less structurally firm, but it could still do the activity without all the aromatic amino acids. Actually, in this case, it helps to stabilize the structure with ligand binding. So where are we going with this? We did a few more studies, and taking all of this together, we think that yes, proteins can totally do with less than 20. Even the 10 early amino acids are sufficient, at least in some examples. Of course, it is a much harder job. And what we would like to do next is we would like to look in a more in vivo environment. We would actually love to see if we can recover whole metabolic pathways in some species with limited amino acid sets. And we would love to, if a whole cell can cope with maybe a lack of some amino acids. And that's why we started talking to you, John, and to you, the minimal cell community. The very first experiment we would love to do with the mycoplasma would be the simple evolutionary adaptation to decreasing amounts of amino acids. This is roughly the pipeline. It says auxotroph at the beginning. I know that mycoplasma doesn't need this. The truth is we started training this approach with E. coli, so that's why we played with auxotrophs. We would like to now approach the microplasma with the same logic, try to see which amino acid is actually the one that can be most easily left out. We would like to, of course, observe the mutations that come up, see which substitutions would be most viable if we remove one of these amino acids, and potentially then we would like to go farther and play with the genome and hopefully push it as far as we can go and see first which amino acids and second how far we can go. So this is where we are at the moment. I would like to thank my group in Prague. We have several coworkers, but we're kind of alone on this topic completely. We have John on the phone at the moment, and we are really grateful for this. We're really interested in community here. Especially, we would want to know if anyone has like some good recipes for defined media for growing the JCVI strains. Anything that can help us get started would be really appreciated. So that's all from me, and I'm unsure about how to here stop sharing. Okay.
Carla, I am so psyched. This idea that, already we have a couple of instances in our minimal cell where I think in some ways unintentionally we have used it to recapitulate what aspects of primordial cells may have looked like. The absence of bacterial histone-like protein or the lower concentration is one, and then the absence of some of the cell division proteins makes the cell divide differently. But it certainly seems reasonable to me that primordial cells didn't have all 20. I was surprised that when you're with your coli efforts, you didn't start off by trying to get rid of cysteine.
We are scared of cysteine because it's just so unique and so unlike anything else. So we are trying to choose mainly those amino acids, which first were the latest ones added to the code, at least according to the available studies. And second, those that actually have some similarity to other canonical amino acids. So that's the logic we're using at the moment. Cysteine is, of course, very sort of tempting to approach, but we have not done it just because we think it's so unique and would be hard to find a good substitute for it. So we're trying to make it easier for us, but we're just at the beginning, so maybe one day.
But in the bacterium, there should be no disulfide bridges, right?
In E. coli?
Well,
in general, in bacterium. Sure, but it's just a very prevalent amino acid at active sites. It's important for metal ion coordination. And at the same time, whenever it's already there, preserved in some active site, it seems it's very much conserved. So I think that. And at the same time, there is a big debate about cysteine and its prebiotic availability. There is some sort of contradiction. There are other amino acids which seem to be more agreed upon. So we tend to focus on those. It would be fun to do all 20, of course. It would be great to, just have a pipeline to test the indispensability of all 20 at the same time. But You're at the beginning here, yes.
Cells without proteins entirely, lipid enzymes. I like it.
I hope not. I actually do believe in peptides and simple proteins a lot. So I think, and I think they have been around from the very beginning, just because they are so omnipresent. They're on meteorites. Amino acids are found on meteorites. They're very easy to polymerize. So it's just it's hard to imagine for me a cell without at least peptides there.
Anyone else? Questions for Klara?
Hi, this is Troy. I just had a quick question, kind of related. So you're talking more about proteins, but have you ever thought, this is kind of a thought I've had, is like the same thing can kind of be done or thought about with like the nucleobases, and that we had 4 of them. Have you ever thought in terms of, would you be able to like remove a nucleobase? Obviously with 4, you're able to make a certain number of combinations to make the codons, to make the proteins, but could this diminishing or this reducing of the number of amino acids also allow you to reduce the number of nucleobases you have and still be able to make a variety of codons to code for those to code for those amino acids?
Yes, that is beautiful. And sorry, there's this echo again. Anyone knows why that is? Oh, it's better now. So that is beautiful, and there are also some hypotheses about a simpler alphabet on the nucleotide side that would come along with the amino acid alphabet evolution. We have not really thought about, doing this ourselves. I think there are more people actually working on the sort of DNA alphabet than there are people working on the protein alphabet. So, and I also feel like those are very differently, they're also very different in terms of approaches. So we haven't done it, but I would love if somebody did this along. In general, I feel there has been more done on the nucleotide alphabet, also, finding alternative alphabets. There is like at least 3 groups working on this. So I think that there's already being shown a great flexibility on the DNA alphabet. So I believe it would be possible.
Okay, thank you.
I see a question in chat. I'm Dayushi Takahashi, a postdoc in Makoto Miyata's group at Osaka Metropolitan University. Is it possible to construct membrane proteins which, in my memory, tend to possess basic isoelectric points only with ancient amino acids?
Yeah, that's a good question. So yes, we think that generally the acidic proteins compensate for the lack of positive charge by using a lot more metal ion interaction. And we think this is how this would easily be solved. I think that already membrane proteins often interact with the phospholipids through metal ions. Probably not all of them, but if there is an interaction between a acidic residue, then there is this metal ion bridge involved. So we think this is how that would be compensated. Daiichi, and thank you.